This is one of the modern pacemaker that we have in the market, and this is the needle which goes on in the interval wall of an heart and paces through the electricity through this needle, and this is the battery part of it. And we use it currently, but every 6 to 7 years this battery is depleted, so we need to change it. To replace this technology, very bulky and planar and sort of expensive technology, we come up with another type of device which is purely flexible, and we have a piezoelectric layer on top. So maybe to open a little bit up, the piezo means stress-strain deformation, and electric means positive and negative charges. So when you mechanically deform this piezoelectric thin layer on the top of a plastic substrate, you create positive and negative charges, which can be buffered through a rectifier connected to the piezoelectric module that we have, and then it goes all the way to a rechargeable battery. So either you can just use the electricity that you produce from the device, or you can charge it, you can store it in a rechargeable battery and use it whenever you need it. So with this technology, for instance, we did a preclinical studies on cow, sheep, and pig heart, and we can recharge this rechargeable battery in 3 hours on a beating heart, a normal beating heart, which can be last for 7 years. So you technically double the lifespan of a pacemaker technology that you have. In addition to wearable devices, we also work on three-dimensional devices like a needle type, and with these devices we try to explore in deep brain of animal models to understand the underlying mechanisms of Parkinson's disease and neurodegeneration. Currently, if you are a Parkinson patient, you need to take the drugs orally or intravenously, which is unnecessarily affecting your whole body. And plus, for some particular drugs, you have to pass the blood-brain barrier. But with this technology, we chronically implant this device in different animal models. For instance, for rats, it's a small brain, it's a fairly small device, shorter. This is for non-human primate, it's a little bit longer than the rat model, almost 10 centimeter. And we have multifunctionality inside. For instance, we have a 20 micrometer diameter infusion cannulas for 2 different distinctive drugs that we can infuse with the pinpoint accuracy. And also we have a homemade electrode system which can measure the neuronal potential while you are infusing the drugs, kind of a closed loop system. So we read out the neuronal potential, and according to that we infuse drugs, or with another layer infusion channel we infuse Silene or ACSF to clean the effect of the drugs that we just infuse. So we can do different regulations, neuronal regulation and modulations inside the brain, in addition to electrical neuromodulation that we could do with this single device. And the diameter of this entire device platform is as thin as your hair fiber, around 150 micrometer, and we have multiple channels inside. And not only for the brain, this device we think would be very valuable, especially for pharmaceutical companies, to test the effectiveness of their drugs in the deep brain or any other organs which are hard to be reached through intravenously or orally. What we want to achieve with this device technology is to do multiple neuromodulation at the same time, to have a better treatment strategies. For instance, right now we do DBS in the deep brain, but not necessarily targeted drug delivery at the same time, to find a different treatment modality for patients, for doctors, and as well as we have an electrode which can read out the neuronal potential while you are doing all this neuromodulation inside the deep brain. So we really would like to investigate and study how the neurodegeneration is progressing while you are infusing a drug, and at the same time while you are doing deep brain stimulation, and while you are reading neuronal potential simultaneously, to find all the variables together and come up with a new way of treatment for the patients.